Hey guys, my name is Chad Pecot. I am the director of the UNC RNA Discovery Center, and we're really excited to be able to host two awesome speakers. First, want to just thank the RNA Collaborative. It's a really cool organization and shows how small a world we are. I also want to thank Grace for hosting. She does an awesome job organizing this, so thank you, Grace. The first speaker I'm going to introduce is Alain Laederach. His presentation is titled Pre-mRNA Structure Change in Controlling Alternative Splicing. And I just want to share one fun fact about Alain, which I did not know, is he is apparently an expert at remote-controlled piloting aircrafts, including airplanes and helicopters. So I don't know if he has any slides on that, but probably not. And we'll take a 5-minute Q&A after his presentation, and then before we move into Mauro's presentation. All right, Alain, it's all you.
Thank you, Chad. So I'm going to stop my video, share my screen, and here we go. And play. All right, so I changed my title a little bit and I shortened it. And I want to caveat this presentation. I am going to talk about pre-mRNA structure to say everything I'm presenting today is still unpublished. And so we're working on this story and we haven't quite finished it. But what I hope to achieve today is to convince you that pre-mRNA structure is interesting and important. So, before we talk about pre-mRNA, we should talk a little bit about mRNAs. And of course, an mRNA, as most of you know, has to have a 5' cap, a 5' UTR coding exons, and a 3' UTR and a poly A tail to be translated. Now, before that, in most higher eukaryotes, there are these really long introns that are transcribed as well, and these are spliced out by the spliceosome. And so that's an interesting and highly regulated process, and it's something that we're beginning to be interested in my lab as well. So before we delve into sort of the data and so forth, I wanted to kind of remind you of what our pre-mRNAs, what human pre-mRNAs really look like in terms of length. So on the x-axis here are length in nucleotides of 5' UTRs, exons, and introns. And you can see that on average, a human mRNA has two 5' UTR exons. Remember, exons don't have to be coding. And they are median length 99 nucleotides. We have 9 on average exons, and they are median length 122 nucleotides. But the real elephant in the room here are these introns, right? And introns have a median length in our genome of about 1,800 nucleotides. And then, of course, there are 3' UTRs, which are quite long as well. And we have 9 of these per mRNA. So really, when you break it down by percentage, about 90% of pre-mRNA is intron. And so they're really kind of the elephant in the room. And I also wanted to put two sort of statistics on this particular slide. And one of them is, this idea that the Pol2 rate of transcription, there are many different estimates of this, but I chose this number, about 3.8 kilobytes per minute. Means that your median, your average intron takes about 30 seconds to be transcribed. And that's actually quite a long time in terms of molecular scale. Because biophysical studies of RNA folding, of RNA secondary structural formation, this sort of initial electrostatic collapse, whether by single molecule pulling or by different time resolved techniques, had concluded that secondary structure formation occurs on the sub-millisecond to microsecond time scale. And these are pretty conservative figures. So that basically means that the transcription during which an intron gets transcribed, the time it takes is really an eternity with respect to RNA secondary structure formation. And so there's really sort of this disconnect in terms of time scale. It's maybe not a disconnect, but we can't completely, just based on these numbers, ignore the fact that introns may have secondary structures. So one other thing that I wanted to mention about introns that's really interesting about human introns is that they are chock full of repetitive elements. Now, there are other repetitive elements, but two of the major ones that we think about are line and alu elements. Now, these are ancient, usually retrotransposons that have been deactivated. And so a retrotransposon is basically an element that can turn into RNA and can reinsert in the genome somewhere else. So it has a reverse transcriptase and has all of the mechanism to be able to insert its DNA into the genome. And that's where they're from. And the major ones we're going to think about today are line and in particular ALU elements. ALU elements don't encode a reverse transcriptase, but co-opt a line reverse transcriptase to jump around. And there's about 1 million copies of these ALU elements in our genome, thus comprising about 11% of our genome. So this is really a major part of our genome that we don't fully understand the function or lack of function of. And the other thing to think about is that it's estimated that about 5% of new births have a novel ALU insertions. So some of these are still active as well. Now, repetitive sequences have always been a challenge in terms of genomics, just simply because if you don't have long enough reads, you really have challenges of where to align reads. So if a sequence is repetitive, you don't know whether to align it to the red, green, or blue, especially if your sequencing accuracy isn't too high. But fortunately, very recently, the genomics community has finished this. It's pretty famous, I think, the telomere-to-telomere genome. It made the cover of Science. It even made the New York Times. But this was really a very, very careful sequencing of one individual's genomes from telomere to telomere, where there was particular focus on these repetitive sequencing using very long read sequencing. And so this sort of opens up a field now where we can really sort of study these repetitive sequences at much larger scale, thanks to this development as well. So if we look in the telomere-to-telomere genome, you can see that within introns, about 13% of introns, which is what we expected, are these ALU elements. But the distribution I really want you to look at is this one on the right. And this is the percentage of the intron that is an ALU. And you can see on average, maybe the median is around 10, 15% here. But this distribution has a really long tail. And there are certain introns, certain of our introns, that are up to 80% ALU, repetitive element, right? And that kind of bothers me. I'm kind of like, how is this possible? How is this functional? And so, if you look about, and this is really from an RNA structure perspective, is that if you have two ALUs, and obviously ALUs can insert either into the positive strand or into the negative strand, but they're always going to be transcribed in the same directions. These ALUs can form base pairs, right? In other words, they have the potential to form secondary structure. Secondary structure formation is very rapid as well. And so you may think of an intron, right, at like this, like, oh, this big, sort of spaghetti molecule that has, maybe small amounts of secondary structure. But in fact, there are certain introns, and this is a real human intron that's probably currently being transcribed in your genome somewhere. This is in the BRCA1 gene. That's up to 80% intron. And when you predict its secondary structure, this is a very small slide, you see a huge amount of secondary structure formation. And this bothered me. And this was really something that there just shouldn't be this much base pairing potential in any RNA, because we know that double-stranded RNA elicits all sorts of immune responses in the cells and so forth. So how is a molecule that has a pretty significant lifespan doing this, and what's going on, and what's the thermodynamics of this, and what's the folding of this? And that's really what I want to talk about today. So I figured if we were going to study these introns and these secondary structure formations, we should really have a functional readout. And one of the functional readouts that is interesting to look at is splicing. So again, this is showing actually to scale a particular region in our genome where you have these 3 exons. You can see how small they are and how big the introns are. And normally these 3 introns are spliced out and form 3 exons in a row. But obviously, sometimes what we know is that this splicing can either be defective or so forth, but instead of the 5' splice site splicing into the 3' splice site over here, you get back splicing and you get the formation of a circle. And it turns out that this mechanism is often mediated by these long-range base pairs. And of course, circles have different functions as well, but at least this is a function we can read out. And so we went back to the literature and we decided we needed a model system to look at this. And this is a model system that has been studied by the Willis Lab. by many different labs as well. And so we knew that it was a good system to start with. And this is the HIPK3 exon. It's a very long exon in terms of like exons. It's about 1,099 nucleotides, but it has these two allo repeats. One is the upstream one is in the negative orientation. The downstream one is in the positive orientation. And you can see that they have the potential to base pair. And the idea is that this base pairing can help facilitate this backsplicing reaction. So just to show you that this is the case, we've designed several constructs. So this is a wild type construct. And we deleted the 5' allo, we deleted the 3' allo. We just wanted to make sure we could get an assay to work with this. And we just deleted both allos. And when we transfect this into cells using a plasmid reporter system, we can actually design PCR probes, QRT PCR probes, across the backsplicing junction here. And this gives us an idea of how much circle there is. And we can also design a PCR probe across the exon-intron junction, which gives us the amount of pre-mRNA. And so in this way, we can measure circularization efficiency, not with respect to downstream splicing, but with respect to total amount of pre-mRNA present. And so when we do this on these constructs, not surprisingly, this was known in the literature before, the wild type construct circularizes. You can see we get significant abundance. But when we delete either the 5' allo or the 3' allo, or both allos, we eliminate circularization. And so this gave us confidence that these allos, as others had reported, can form these, are essential and depend, the circularization reaction depends on both these allo elements being present. So the idea that they base pair is one mechanism. And so I have an artistically minded student in the lab, Colin, and he's made these nice graphics. And so the model here really is that the upstream allo and the downstream allo might be forming base pairs, and this might be facilitating bringing this 5' and this 3' together. But in reality, we had never really measured this base pairing. Now, I don't have much time today, but I just do want to show you about this experiment. My colleague Kevin Weeks and I developed this technique called shape jump. And the basic idea here is to use a shape crosslinker. So for those of you who don't know, a shape reagent acylates the 2' hydroxyl of an RNA. And in this case, we're using a bifunctional one. And in so doing, we are able to cross-link and find nucleotides whose backbone are close together. And we use this strategy of a very special, highly processive RT that instead of stopping at the cross-link, jumps across the cross-link. And so if we sequence these reads and we measure deletions in them, we can know that these two regions of the RNA are close together. So using clever priming and a bunch of sequencing, which I don't have the time to go into, we were able to really show that these long-range interactions occur in this pre-mRNA. And you can see these jumps are occurring. And what's interesting is that they're actually occurring not necessarily across secondary structure, but across these large loops of unpaired nucleotides, which is what we'd expect. We also see quite a bit of jumping at the 5' and 3' end of the base pairs. So with that, I kind of need to introduce one more concept here, and I've alluded to this before, right? Is that for base pairing to occur, you need an allo that's inserted in one orientation upstream and a different orientation downstream. And we call these PALUs, which we think is a cute name for them because they're kind of pals, because they can form base pairs. But if these two allos upstream and downstream are in opposite directions, or in the same direction, then they're not going to be able to necessarily base pair. And we call these lonely allos, LALUs. So Colin, who's a very talented student in the lab, helped us develop this measurement at genome scale, which allows us to measure the probability or the odds ratio change if you have a PALU versus a LALU upstream and downstream of an exon. And fortunately, because the human genome, we have the T to T genome, and we also have a very large amount of RNA sequencing on it, we know across many, many tissues where there are circles and where there's not circles. So we have a very large N. And so this allows us to do this this calculation, and we can see that the log's odds difference ratio of circularization versus not is higher in PALUs versus LALUs. This is statistically significant. But again, I want to show you the scale here. This isn't even an order of magnitude difference. So yes, this base pairing interaction seems to be driving some circularization, but it's not everything and the endpoint to everything. So if you're trying to build a predictor based on this, it wouldn't be a terrific predictor of base pairing. So with this, and as I mentioned, this is still a work in progress, we have to think a little bit about ALU families. So ALUs don't obviously have different sequences. And there are three large ALU sequences in the human genomes. They're called J, S, and Y. And the mnemonic that we use to remind ourselves is Y are the youngest, and S are the smidlest, and J are the oldest. So this is not the perfect mnemonic, but at least if you remember Y is the youngest, these are usually the most active ones. But others have classified ALUs into many different subfamilies. And so we wanted to know if this subfamily classification would help us better understand whether there's circularization or not. And just to convince you that these are distinct, if we do a dimensionality reduction on these different ALUs, these are across the T to T genome, you can see that regardless of which dimensionality reduction we use, you can see that there are these different families, ALU-Js, ALU-Ss, and ALU-Ys, and that they cluster quite well. And that depending on which clustering you use, there are even very distinct families that you can separate out further. But I wanted to go back to our HIPK3 example here and show you sort of the probabilities, because HIPK3 is not the only example of an ALU-SZ type ALU that's upstream of an ALU-SQ. In fact, there are hundreds of them. I asked Colin to include the numbers in here. This gives you an idea of just how common this setup is. And what we can see when we set this up this way, here we're looking at the logs odds ratio. Again, up here are the 2+ orientation on the upstream, and down here are the 2- orientation on the downstream. So these are PALUs in these two corners, and these are LALUs. There's definitely a difference between red and blue, between PALUs and LALUs. The LALUs are more blue, and the PALUs are more red. But you can also see that between different families, for example, ALU-SQ2+ versus ALU-SZ is much more likely to circularize than, say, ALU-SC+ to ALU-SQ-, even though those are two PALUs. Similarly, in here, there's differences in the LALUs as well. So the simple sort of idea of sequence similarity is not a great predictor of whether PALUs can circularize or not. And so I want to go back to some of the data in our HIPK3 example. And I want to say we didn't just do shape jump on this. We also did a bunch of different chemical probing reactions. And I want to sort of show you what happens when you make a prediction of RNA secondary structure without data and with data. So you can see what you generally see is that when you include data in the RNA secondary structure prediction, which is shown whether this is DMS or shape data, you can see that in general, there's less base pairing in the predicted structures when you include these data in there. This is something that's well known about RNA structure prediction. But it does mean that if we just use thermodynamic models, it seems that we're overestimating the base pairing or the free energy of formation. And so this made us think, in fact, is circularization somehow driven not by perfect complementarity, but are there specific aspects of RNA structure that are not allowing it to form these perfectly, even though they're perfectly complementary, these perfectly base paired interactions. And so I just want to show this slide to show you that this is statistically significant. If we use the DMS data, the experimental data on no data, on a model that's folded just with M-fold, if you like, we don't do as well as if when we inform with the data. And this really tells us that this opening up is an essential part and that the experimental data is suggesting there's less base pairing than what we observed by thermodynamic prediction in ALU-ALU interactions. And so if we now go back to the whole genome and to every possible ALU family, these are all the different ALU families, Again, it's organized in the same way. You have LALUs here, they're more blue, and PALUs here, they're more red. But what you'll see is that there are very red dots in here, and there are very blue dots in here, or squares in here as well, right? So there are certain LALUs here, plus orientations, that actually circularize quite well. And there are certain PALUs that don't circularize very well at all, such as these squares as well. And so we were interested, especially in the PALUs that don't circularize and the PALUs that do circularize. And we wanted to see if there was a difference in the distribution. And so what I want to show you here is a new kind of way in which we're measuring. We're basically just aligning the two ALU sequences in frame to determine their base pairing potential. This isn't a free energy of folding because we think that that overestimates base pairing. And we wanted to see if we could measure a difference between PALUs that form circles and PALUs that don't form circles. And you can see the medians of these aren't different, but N is so large that, again, this is statistically significant. But I really like things where I don't have to show you a p-value, but where you can just sort of see the difference. So what if we now go back and we look at one of these really blue squares? So these PALUs that aren't very circle forming. And you can see that's this distribution that I've shown you here. And what's interesting with this distribution is that it's very much shifted to the right, to sort of high base pairing potential, right? So it's even further to the right than the median of all the genome. If we now go back and look at the base pairing potential of a really red square, a red PALU, right? You can see it's actually not shifted to higher base pairing potential, but more to the left, to lower base pairing potential. And so as a result, we are concluding, or we're thinking, that it is quite possible that, in fact, it's not really the ability to form base pairs. It's not the most thermodynamically stable PALUs that circularize, but in fact, that there needs to be some flexibility in these base pairing reactions. So as I mentioned, this is not a complete story, and I have 30 seconds, so I will end there so that Mauro has plenty of time to go. And I will finish by acknowledging Dr. Justin Taylor. Justin came to me from Marlene Balfour's lab in Albany, and he was an expert in group 2 introns, so in self-splicing. So when he came to the lab, he was really the one who introduced us to ALUs and started talking about retrotransposition. So I thank him for that. You saw a lot of work from Colin Taylor, who did a lot of the bioinformatics, and as well as work from Scott Allen and Claire are both postdocs in the lab who are also working on this project. And Anastasia did some of the initial overlap as well, and Abby's also working on that. I couldn't have done this without my collaborators, Kevin Weeks, Sylvia, Dave Matthews in Rochester, Rolf, who just came to do a sabbatical in our lab. And of course, Bill was the one who first told me about ALU elements and backsplicing. And so can't forget about him. Funding, and this is the fantastic lab. And with that, Chad, I guess if there are any questions, I'm happy to take them. Otherwise, we can move on tomorrow's Q&A talk as well. So thank you.
Alain, awesome talk. Thank you so much. I do have one question right here to start with, and if I have time, I'll maybe ask one myself. So the first question says, you showed a regression of the impact of I-ALUs and P-ALUs on circularization. Are there other factors known to be associated with circularization beyond ALUs?
So yeah, that's an excellent question. So let me just stop sharing. So anyway, okay, I won't do that. So yes, I mean there are many factors that seems DHCF2 is one. There's an RNA binding protein that seems to be actually binding to ALUs as well. The length of the exon seems to be something important. You also don't want too strong of a 5' splice site, because you have to remember that RNA is co-transcriptional. So the strength of the 2 splice sites has to be sort of in between again. But it seems to be that it's kind of, to form a circle, you need to sort of be in the median. You don't need to have extremes. You don't need to have super base pairing. You don't need to have super strong splice sites, because otherwise those will just splice normally. So it seems to be a balancing act. So yes, there are many, many different things as well. Yes. So that's good. So yeah,
Lots of good questions coming in. I'm just going to ask them in the order they come in as time
sounds
allows.
good.
Next one is, how do you deconvolute locations where there are three closely spaced ALUs, say two on one side, one on the other, where the P-ALU and L-ALU distinction is optimal?
So we take the closest one so far because we need to simplify. Again, a work in progress.
Yeah, great. Hi, Alain, great talk. For your structural commentary on the 5' and 3' ends, is this akin to what Dave Matthews and Dmitry Ermolenko were saying about mRNAs and lincRNAs having short end-to-end distances at the 5' and 3' ends?
Yes, I think so. Although these particular constructs we're studying, right, have a huge base pairing potential on 5' ends and 3' ends. I think, this concept of secondary structure distance, which is the context here, is very important, and that is a very valid point. That's what's going on here. But if you think about it, it's not really the end. It's really the 5' and the 3' splice site, which is more internal, which need to come close together to allow back splicing. So.
Great. Next question. How is circularization related to the number of ALUs in the introns or distance from the junctions?
So that's a good question. So distance from the junctions, we're looking at that right now. Again, we're looking just at the proximal one. It gets more complicated if you start looking at 2 or 3 and so forth. So Bill Marsloff and Aravind Esekant had a great paper where they showed that I think if you move the 5' ALU closer, it circularizes more. The 3' ALU further, it circularizes less. There's really interesting distance things as well. We haven't really seen yet a super strong signal in the genomic context yet to that. So we haven't figured that out.
Great. They're coming in fast, so I'm just going to read them fast.
Okay.
What about the processivity of splicing, and how might that restrict different introns from interacting with each other?
Right. So obviously, if you splice out the upstream the upstream intron before you splice out the downstream, and before this downstream intron is fully transcribed, you won't be able to make that circle, right? So definitely, again, having a strong 3' acceptor site upstream is really important if you don't want circularization. So that's, I think, how that's balanced out as well.
This is an interesting one here from Norman Chu. Do RNA modifications interfere with back splicing?
So probably we don't know yet, and I think that's just another complicating factor in all of this as well, right? I mean, you can imagine all sorts of modifications trying to keep these ALUs from base pairing, right? But that has to happen fast because again, RNA secondary structure formation is very fast.
Cool question, Norman. Dan, I'm going to let you ask Alain your question later if you don't mind. I got one last one. Do we know what the purpose of circular exons is? Are these just mistakes? Are they regulated?
That is an excellent question. I will say no comment on that because I've shown that there is evidence that they act as microRNA sponges, but I suspect we're going to find many other functions for them as well. So thank you.
I would agree with that too.
Thanks.
All right, that's all the time we have for questions. A lot of great questions. Sorry we couldn't answer all of them. I want to make sure I have time to be able to introduce Mauro so he has time to talk as well. So Mauro's got a really cool title and really intrigued to see what he's going to share. So I'm going to read it off. I don't know if he's changed it as well, but the concept's really interesting. So ectopically expressed, Airn lncRNA recruits Polycomb chromatin without inducing gene silencing. So that sounds intriguing and non-canonical. One fun fact about Mauro is he said he's a big fan of the Durham Bulls. I told him I didn't know there was a big fan. So this is one of them. Here we go. It's all yours, Mauro.
All right. Thank you, Chad. Thanks to the organizers, Grace, and everybody else involved, and thanks everyone for coming today. So as Chad mentioned, my name is Mauro Calabrese. I'm an associate professor in the Department of Pharmacology and the Discovery Center here at UNC. And speaking really broadly, actually another one of my fun facts is I love North Carolina barbecue. I wasn't sure which one to provide, so but here it is on this slide. Speaking really broadly, research in my lab aims to understand how gene expression is regulated. And this is a really fundamental and complex issue in biology. Humans, for example, are comprised of hundreds of cell types and trillions of cells that all need to work together in real time to deal with a constantly changing environment. And I'm going to actually try and open up my thing here because I know I'm just going to stop my video, which is what I was instructed to do. So, so yeah, just as an example, humans, a lot of cells, we need to function. Our environment's constantly changing. And, at the core of this dynamic response to our environment is a dynamically interpreted genome that decides what genes are expressed, when, where, and in what quantity. And not surprisingly, given its fundamental importance in biology, when genes and genomes are misregulated, disease frequently follows. And of course, there are many examples of this, but I think cancer is a very good one where misregulation of a handful of genes in a handful of cells can ultimately give rise to a fatal disease. But not only can altered gene regulation cause disease, it can also be harnessed to treat or cure it, as well as advance health in many other ways. And this is just to say that understanding the fundamental mechanisms that govern gene regulation, not only in our own cells, but also in other organisms, remains a really important endeavor. And so within the broader umbrella of gene regulation, research in my lab aims to understand how gene expression is controlled inside of the nucleus by molecules called long non-coding RNAs. And before we get into specifics, I want to just reflect briefly on the DNA content of our genome. As humans, our genome is comprised of 22 autosomes and 2 sex chromosomes, the X and the Y. We're diploid. In each one of our cells, there's about 6 billion base pairs of DNA, amongst which are distributed 2 copies each of some 20,000 protein coding genes. And proteins are really the biomolecules that do a lot of the work inside of our bodies. But despite the importance of proteins, I think it's interesting to recognize that only a small fraction of our genome actually encodes for protein, about 1%. But upwards of 80% can and does produce RNA in different settings. A full 20% of our genome presently is annotated as a long non-coding RNA, which are molecules that have little or no potential to encode for protein even after they've been spliced. And I love giving this talk after Alain talked because I also think it's very interesting that such a large fraction of our genome is comprised of an intron. And as many of you know, introns and also enhancer RNAs and others, RNAs produced by our genome, they tend to be very lowly abundant. They're considered to be unstable, and most of them never leave the nucleus. And I think it's a reasonable question to ask, looking at this data, why? Why do our genomes make so much RNA if most of it doesn't leave the nucleus and is going to get turned over shortly after it's produced? And of course, there are many answers to this question, but we would like to think that at least part of the answer is that some of this RNA may play important roles in controlling transcription. And it's in that context that I'd like to talk today about a long non-coding RNA called Airn, antisense to IGF-2R non-coding RNA. This is a very interesting region of the mouse genome. The Airn gene is, depending on the annotation, anywhere between 90 and 120,000 nucleotides long. It produces an RNA that can reach 90 kb, although it experiences what we believe to be premature transcriptional termination across its length. The Airn non-coding RNA is also almost entirely unspliced, which is really rare for an RNA of this length. You can see by RNA fish here in yellow that the Airn RNA is retained at its site of transcription. If we try and measure its overall abundance using quantitative RNA-seq, we see that it's present at about 9 molecules per cell. And by actinomycin D, it's rather unstable. So the half-life of this non-coding RNA is about 1 to 1.5 hours. And again, Alain mentioned this, but the rate of polymerase II transcription in mammals is anywhere from 1 to 3 kb. So it might take about an hour to produce a 90 kb long non-coding RNA, and that's about its half-life. So really low abundance, fleeting RNA. It's also not even conserved outside of rodents. Nevertheless, it has this really amazing function. It's required to repress gene expression over about 1/10 of mouse chromosome 17. And it does so in an imprinted manner. So for those of you who haven't heard of genomic imprinting, this particular non-coding RNA is only expressed from the paternally inherited chromosome. and that is the chromosome that it induces gene repression on. An important aspect of repression by Airn is its ability to recruit repressive histone modifying enzymes called the Polycomb repressive complex 1 and 2, or PRC1 and 2, over its target domain. If you've never heard of them, the Polycomb enzymes are really important for initiating and or maintaining gene repression in many regions of the genome, including this region that I'm showing you here surrounding the Airn non-coding RNA. And I want to just walk through what some of the Polycomb data look like, just to give you a sense of how impressive the biological function of this RNA actually is. So you can just focus your attention on these top two plots here. These are tiling density plots of allele-specific ChIP-seq data for the two histone modifications deposited by Polycomb. So K27 trimethyl, doesn't matter the name. This is PRC2 dependent, and this is PRC1, is K119 ubiquitin. The data are split by allele. So the Polycomb modifications on the paternal allele are shown in blue, and those on the maternal allele are shown in red. And the higher the dot is on the x-axis, the more Polycomb-directed modification there is in that region of the genome. And what we've done in these plots is we've centered the Airn non-coding RNA locus, and you can see, on the paternal allele specifically, this really incredible enrichment for PRC1 and PRC2 directed chromatin modifications, over what ends up being about 15 million base pairs. And we see something very similar if we profile the catalytic components of both PRC1 and 2 by ChIP-seq. We know these modifications are Airn dependent, but here's just a way to convince you of that. So if we truncate the Airn non-coding RNA just downstream of its transcriptional start site with a triple polyadenylation cassette, you see that all this really incredible enrichment of Polycomb on the paternal allele drops to baseline. And more than that, we can overexpress the Airn non-coding RNA from its endogenous promoter using catalytically dead Cas9 fused to a transcriptional activator. And you see this really incredible increase in Polycomb-directed modification, again, specifically on the paternal allele, which expresses Airn. And the last piece of published data that I'll show you today, we can also map regions of chromatin that are proximal to the Airn non-coding RNA using an approach called CHART. If you've never heard of CHART, it's analogous to ChIP, but instead of using an antibody to retrieve a protein and its associated DNA, we're using antisense oligos to retrieve the Airn link RNA and the DNA that it's near in formaldehyde cross-linked cells. And you can appreciate this top panel is our CHART data. And if I didn't tell you that these were CHART, you would have confused them for ChIP. The CHART signal are really incredibly colocalizing with all of our Polycomb ChIP data. And so, it's really clear that the presence of this non-coding RNA near chromatin leads to very robust recruitment of multiple forms of Polycomb enzymes. But as I mentioned earlier, this is a really unusual RNA. It's lowly abundant, it's unstable, it's unspliced, it's imprecisely produced, and it's not even conserved outside of the rodent lineage. More than that, probably PRC1, but definitely PRC2 are known to bind sequence motifs that are relatively common in RNA. And so mechanistically, it's very much unclear how this RNA, as well as others, recruit Polycomb to chromatin and induce gene repression. And in this particular case, it's even been questioned whether the RNA itself is functional, or is it something associated with perhaps its active transcription in this region of the genome that's leading to these really incredible phenotypes. And so the question I'll begin to address now in the coming slides is, to what extent is Polycomb recruitment in this region of the genome driven by Airn versus features that are inherent to this region of the genome, including the act of Airn transcription? And to begin to address this question, what we thought we would do is take the Airn gene and put it somewhere else in the genome and see what happens. And so working with some really talented molecular biologists here at the UNC Animal Models Core, we were able to achieve just that. So we cloned a doxycycline-inducible version of the Airn non-coding RNA gene into a specialized plasmid and used some sort of tricks with pre-recombinase to insert that transgene into a single copy locus in the mouse genome on a single allele. So the specifics are probably not so important, but this is a single allele of chromosome 6 in the ROSA 26 locus. We now have the Air non-coding RNA under the control of a doxycycline inducible promoter. And we have previously used this system to study a long non-coding RNA called EXIST. Some of you on the call, I'm sure, are familiar with EXIST, but if you've never heard of it before, it's another really incredible RNA. This is a gene that's expressed from the inactive X or the inactivating X, and the function of this long non-coding RNA is to silence gene expression and recruit Polycomb repressive complexes over the entire 165 million base pair X chromosome. So it has a function that's analogous to Air, but it's even more, it's better at it. It can recruit Polycomb over a whole chromosome and silence a lot of genes. And if you look at, in a natural setting, the abundance of EXIST and the stability of EXIST relative to Air, what you see is that EXIST is naturally about 25 times more abundant than Air, and it's about 4 times more stable. And so, the second question we're able to address with this transgenic system is, how do the effects of Air expression compare to those induced by EXIST expression when these two RNAs are expressed from the same ectopic locus, which is the ROSA 26 locus? And so now I'd like to show you some of our characterization of these new cell lines. So what we find is that when we express Air from the ROSA 26 locus, a lot of its endogenous properties are retained. So comparing the levels of Air induced from ROSA 26 to those of EXIST induced from the same locus, we see that Air is about 12 times less abundant than EXIST, and it's about 10 times less stable. And because we're expressing these RNAs from the same location in the genome, we can conclude that these differences are inherent to the lncRNAs themselves and not features of their endogenous genomic environment. I also want to point out before moving on that we're also able to toggle the expression of lncRNAs in this setting, at least to a certain extent. So if we add 100 times less of the inducing agent in this system, so we're inducing the expression of these RNAs by adding doxycycline. If we add 100 times less doxycycline to our EXIST expressing cells, we can create a system where Air and EXIST are expressed to roughly equivalent extents. And looking by single molecule sensitivity RNA fish, what we see is that this low expression of EXIST is not entirely explained by just a handful of jackpot cells. We're still detecting EXIST RNA even at this low induction setting in a good number of cells. And so having walked you through the characterization of the system, I'd like to show you what the Polycomb data now look like. First, I'll show you the 2 controls, our negative control on top and EXIST on the bottom. On the left-hand side here is the intensity of PRC1 directed chromatin modification. On the right is PRC2 directed. And you can see in cells that don't express a long non-coding RNA from ROSA 26, there's essentially no change in Polycomb when we add doxycycline. Compared to when we express EXIST, we see this really dramatic and a dramatic chromosome level recruitment of both PRC1 and PRC2 directed modifications. Intriguingly, when we look at cells that express Air, we very clearly see the recruitment of Polycomb directed modifications. So it's clearest when we look at PRC2 directed modifications. You can see when we express Air, there's this enriched region of PRC2 dependent modification that stretches for about 40 or so megabases, mostly centromeric from ROSA 26. And to a lesser extent, we also see what I believe to be a significant difference in PRC1 directed modifications, although you can probably appreciate that the extent of PRC2 directed modifications versus PRC1 that accumulates is quite different here. And the other thing that I'll point out is that when we look at Polycomb changes induced by expression of EXIST at the low level condition, we see that they're largely equivalent to those that are induced by Air. So, of course, there are some differences here, but by and large, when normalized for copy number, these two RNAs are inducing the accumulation of PRC-directed modifications to similar extents, which I think is pretty cool. Surprisingly, when we look at gene expression changes in our Air-expressing cells relative to our empty vector control, we find none. So there's no significant changes here between Air and empty vector control. So what we think is that most of this Polycomb that gets recruited is happening within intergenic regions or over silent genes, and it's unable to infiltrate actively transcribed regions of chromatin. In contrast, if we look in our positive control, we see that at the full length or at the full expression of exist, we see a dramatic repression of genes across the length of the chromosome. And even at this low level expression of exist, we detect 21 downregulated genes. I think this is a meaningful difference, although it is sort of pushing signal to noise here. So ectopically expressed Air can recruit Polycomb to chromatin. It does not appear to, in this setting, recruit PRC1 directed modifications to the same extent as exist when normalized for copy number, and nor is it able to induce gene repression. So we're going to conclude that this Air RNA is functional, and now we're interested in understanding, why and how it does what it does. And one of the things that I think we've done successfully is compare the sequence of Air to exist to a third known repressive RNA that I haven't mentioned until now. It's called KCNQ1-OT1, or just simply OT1. Like Air, OT1 is extraordinarily long and unspliced, 90 kb. Like both of these RNAs, the expression of OT1 can recruit Polycomb complexes and repress gene expression, in this case, over about a 3 million base pair region of the genome. These three RNAs evolved independently of each other. If you compare them by linear sequence content, you really don't see a lot of similarity that you can make heads or tails of. And so what we did is we took essentially a motif counting approach called Seeker and compared the internal sequence content of each of these RNAs at the level of, in this case, 4 mers. K-mers length 4. And because we're interested in detecting regional similarities and differences, we worked through this. In the end, what we decided to do was split each lncRNA up into sequential 500 nucleotide long fragments and compare, every fragment in exist to every fragment in exist, every fragment in Air to every fragment in exist, and so on and so forth. And when we do these comparisons, what we find is that Air is actually no more similar to exist than we would expect from chance. But Air is significantly more similar to OT1, and OT1 is significantly more similar to exist. And if we go on and look at the regions of Air that are highly similar to exist, we find that those regions are more likely to be similar than OT1 than we would expect from chance. And so what we're interpreting here is that, yes, there are differences between these RNAs. I couldn't tell you what that means, but there's more similarity between them than we would expect from chance. And if we search these shared three-way similar regions for enriched motifs, we find them, and I'm showing you some of the most significant ones here over Air and exist. And the thing that immediately popped out to our interest is this C-rich motif, which is essentially equivalent to the known consensus for a very abundant RNA binding protein called HNRPK. And this HNRPK motif was of high interest to us because in 2017, Greta Pintacuda and Neil Brockdorf first reported that C-rich regions within exist bind HNRPK, and that HNRPK binding is really critical to recruit a specific form of polycomb called variant PRC1 or VPRC1. And this VPRC1 recruitment they proposed is then responsible for initiating this cascade of polycomb recruitment over the whole X chromosome. And I'll also point out that, following from the Brockdorff study, we've also knocked down HNRPK in a cell line that expresses exist, Air, and OT1. And we find that at least PRC2-directed modifications are reduced in the regions silenced by all three of these RNAs. And so, what we wanted to know next was, do our repressive RNAs of interest exhibit any sort of enriched association with either HNRPK, 3 components of PRC1, and 5 separate components of PRC2. And in order to address that question, we used formaldehyde cross-linked trophoblast stem cells that express all 3 of these RNAs, and we performed an RNA immune precipitation protocol. And so I'll show you the data over Air first. There's a lot of information on this slide, so hopefully I can make some of it clear to you. But I think the thing that was most obvious when we looked at these data is that there are regions of the Air non-coding RNA that associate with above background levels of what we infer to be variant PRC1 complexes. And this VPRC1 enriched regions co-localize with regions of the Air non-coding RNA that are also enriched in association with HNRPK. So consistent with the Brockdorff model. And more than that, these same regions that are enriched in VPRC1 are the exact ones that we identify as exist-like from our prior k-mer analysis. I'll also point out that, at least under our conditions, we find very little evidence, if any evidence, for above background association between Air and canonical PRC1 or any of the 5 components of PRC2 that we profiled. And I'll just show you the analogous panel of data in exist because I think it's a lot more clear. You can see that there are these regions within exist that are highly enriched in their ability to associate with HNRPK. Those same regions are also enriched in association with variant PRC1. And we don't find really any clear evidence that any component of PRC2 is enriched above background with exist using this assay. So those are the data that I want to show you today. I'll just highlight the conclusions again. So we'd like to conclude here from our data that the Air non-coding RNA has an inherent ability to recruit polycomb to chromatin, but at least in this ectopic assay, it's unable to repress genes. And what that might mean is that in an endogenous setting, the main function of the expression of this RNA may be to sort of promote a memory of a prior repressive event and not necessarily initiate repression. I will say when we normalized for copy number to the extent that we were able to in this setting, we found that Air recruited polycomb to chromatin with an efficiency that, or with a potency that rivaled exist, which is this really incredibly potent RNA. But when we expressed exist to, subjected both of these RNAs to the same degree of promoter activation, exist was way more abundant and repressed gene expression over the whole chromosome and recruited polycomb over the whole chromosome. And so I think, as somebody that also studies exist, I think we're learning some important things about exist from this experiment, which is that its abundance and its stability are really important for its biological activity. We compared the sequence of Air to 2 other repressive RNAs, and we found certainly there are some clear differences, but also some very intriguing similarities. Mechanistically, we think at least some of these similarities are very relevant. So in particular, the recruitment of HNRPK. But it's also possible that these 3 RNAs are engaging different subsets of proteins that are ultimately contributing to their effects in cells. And then the last conclusion here is that we found very little evidence to suggest that canonical PRC1 or any of 5 components of PRC2 exhibited above background associations with 3 different repressive RNAs, Air, exist, and OT1. But we did observe very clear enriched association between PRC1 and these 3 RNAs, and those regions of enriched association co-localize with an RNA binding protein called HNRPK. And so I think, considering our data in context with everything else in this field, we'd like to sort of, the way that I think about this is that probably these RNAs are recruiting histone modifying enzymes by and large via indirect mechanisms through bridged interactions, both that occur on chromatin and other proteins that might be found in the RNA-seeded condensates. And I'll just leave you with the following thought, which is that, I do think the principles that we've discovered with this non-coding RNA Air are probably relevant to at least a portion of the expressed transcriptome. And we're very interested in testing that hypothesis moving forward and seeing, what extent of the RNA that our genomes produce have the ability to locally regulate transcription. And with that, I want to conclude and thank everybody in the lab. This has been a really incredible collaborative effort. Literally everybody in the lab has been working towards it. Jackson, Aki, Shuang, Mickey, Elizabeth, Sam, Rachel, Ryan, Quinn, and then our collaborators, Dale, Steve, and Kyle at the Animal Models Core. None of this would have been possible without all of these people's efforts. I also want to thank our funding sources, the organizers, and thanks again to everyone for attending today. And I'd be very happy to take any questions.
Very cool talk, Mauro. It's an impressive amount of beautiful data you presented like so well. I'm very impressed. I'm trying to see the Q&As right now. I'm not seeing.
Can I ask one, Chad, real quick?
Yeah, go for it. I'm not seeing any questions popping up right now. Go ahead, Alain.
So Mauro, very interesting. So Airn is like 10 times longer than exist or something, maybe not 10, but like 5 times or whatever, right? So when we think about molarity of nucleotides, it's very different than molarity, and yet it's less repressive, right? So how does that work? Or how many is it because it's lower expressed because it's longer, but are there as many nucleotides? So is the per nucleotide effectiveness about the same?
Yeah, that's a great question, actually. I haven't thought of it in terms of molarity, but you bring up an important point. There's like 5 times less real estate in exist. And so, when we normalize it for copy number, Air is probably actually not nearly as efficient as exist is for that exact reason.
I was going to ask a little bit different question, but somewhat related is it looks like you notice about a 10-fold difference in stability with exist over Airn. Do you think that's also related to why you're seeing about a 10-fold difference in abundance with docs?
Yeah, I think it's really, really closely linked. And like Air, exist I think has evolved to be, really precisely produced. And it's possible that, it's splicing, it's got a long poly A tail. There may be some important structures embedded at the 3' end. And Air doesn't really have any of that. It's like kind of like it terminates along its length, and it's probably evolved to be less stable so that it doesn't do what exist does.
I'm not seeing any questions popping up. It looks like we're coming up on the hour, so that's probably tightly related. Thank you both guys. Fantastic talks, Alain and Mauro. It was really, really exciting, really interesting data. So, and thanks also again to the RNA Collaborative. Looks like I actually have one question that just popped up, and I can ask real quick, if that's okay, Mauro.
Sure, yeah.
A lot of your enriched motifs in the lncRNA seem to be repetitive stretches of single nucleotides, A-rich, G-rich, C-rich, U-rich. It's an interesting trend. Do you have any insights into why these types of motifs might be enriched in these lncRNAs?
Yeah, that's a great question, Scott. I don't, but it may be something that is meaningful. I know, if Dan's on the call, but just from his work that there's a lot of motifs, or there's, basically a limited subset of motifs that RNA binding proteins tend to interact with in single-stranded regions. And I don't know, like, if sort of our homopolymers here are a function of the way that we've set out to detect the motifs, or if it's a biological trend. I'm not sure.
Yeah, Dan just had an interesting question as well. Do you think that there is coevolution of the regions they silence in the RNAs themselves?
Yeah, that's a great question, Dan. I do think so. We didn't go into that today, but I think, the X chromosome, there's clear evidence for that. And also in the Airn region, it's an extraordinarily interesting region of the genome naturally. And we think there's a lot of chromosomal folding that is meaningful. So I think it's totally likely.
I think that's all we have time for. I just want to thank again the RNA Collaborative and Grace for helping organize this. It was really cool, and looking forward to watching it again.
Thanks, everybody.
Lots of claps and thumbs up virtually.